Hey everybody, welcome to this very special webinar co-hosted by Angel Bayari from Keybeam. And Keybeam is a clinical research imaging analysis vendor. And actually, they have a lot of interesting technologies that they're implementing. And if anyone joins late, we have the replay link available. This will also go on the Keybeam's YouTube channel, Keybeam's Facebook page, as well as Keybeam's podcast. And let me pull up the slides. And while I do that, Angel, can you introduce yourself and explain a little bit about what Keybeam is?
Yes, well, hello everybody, and thank you, Dan, for the introduction. It's an opportunity for me to be sharing what we are doing in the field of real-time clinical trials and image analysis. I am Angel. I am an electronics engineer that started working in image processing more or less 11 years ago. I started developing several algorithms in image processing and solving radiology problems. And then we decided to create Keybeam in 2012, by the end of that year. And we focused the company on doing quantitative image analysis, right? I don't know if I can go to the next slide because I don't have the, okay, right, I can. So
Okay, good.
This is my background, no, the one I was mentioning. I did the PhD in image processing, and then I decided to become an entrepreneur. I started to push the company more seriously, full time in 2014. I raised funds and I created a team, and I wanted to solve some problems I identified in clinical research. Mainly, I was working in radiology departments, and I was in the other side of the clinical research. I was at the centers, seeing how it was to send images to core labs and for those centralized imaging laboratories that were simply collecting the images but were not providing any kind of feedback or information to the centers or to the sponsors. And it was not until the end of the clinical trials where the results were released, and the decisions on the drug were made. So I identified some, let's say, areas for improvements or needs that could be covered using our technology, the technology I was developing for radiological daily practice, So I started to work very seriously in the business plan for Keybeam in 2015. I was involved in the biggest accelerator of Spain that is promoted by Mr. Juan Roig, who is a very recognized businessman from Spain. He is the CEO of Mercadona, and he is from Valencia and has an accelerator called Lanzadera, and they are selecting the best startups of the country to help them with the marketing, with several parts of that are elementary for the business. And then that money in the accelerator helped me to create a team and consolidate Keybeam for sure. Later on, we let's say that raised funds in a seed investment round, and then we also became an European Commission company, let's say, because we were awarded with the SME instrument phase 2. So now the company, I would say that is in growth process. We are gaining more and more clinical trials. We are being involved as one of the reference quantitative imaging core labs worldwide. And we have seen how we started collaborating with small clinical research groups, like medical doctors that were developing some retrospective studies or prospective studies with small pharma companies. And now we are being involved in more and more I would say, trials with big companies also. So we are right now bringing this concept of, let's say, quantitative imaging, precision imaging, precision medicine, real-time clinical trials in the end into the clinical research, right? So more or less, the Keybeam's origins come from radiology part, from trying to improve the radiological service that radiologists were providing in radiology departments in order to provide a more quantitative, more objective, more reproducible assessment of images, trying to cover those, let's say, problems that the naked eye cannot detect in the images. So this is how we started with the company, right? And please, if there is any question, feel free to ask, okay? Okay, so let's speak about the focus of our company and our technology, which is the quantification of imaging biomarkers. And what is an imaging biomarker? We are quite used to listen about standard biomarkers like blood biomarkers, serum biomarkers, and different type of biochemical biomarkers that we can measure from different biological samples, right? But we are referring to imaging biomarkers as those parameters that we can extract from medical images after applying the appropriate computational analysis techniques. And for that, we always use computing, we use algorithms, and we use mainly techniques like artificial intelligence or machine learning. And not any parameter that we can extract from any images can be considered an imaging biomarker. In the definition of imaging biomarker, we incorporate the applicability or the utility to become an indicator of a diagnostic, a prognostic, or a treatment response value. So I would differentiate any kind of parameter that we can measure from images from a biomarker. An imaging biomarker is more than that. It's a parameter that is demonstrating a real clinical utility, right? And what can an imaging biomarker tell us? Imaging biomarkers can help us to identify findings quantitatively that are not evident to the naked eye. So, for example, we can have diseases that to the naked eye of a radiologist in a core lab, for example, in a clinical research project with Alzheimer's disease, we can have a case that has no atrophy, for example, in the brain, apparently. But if we quantify the gray matter volume, if we quantify the white matter volume in the entorhinal cortex of the hippocampus, then we can identify, for example, that we have a loss of gray matter in this region, right? And we can only do that if we quantify by computational imaging techniques and computational analysis techniques the findings in the images, and we try to provide a quantitative support to the naked eye. In this case, the biomarker would be diagnostic, right? Because it would tell us that there is an atrophy in the hippocampus. But we can have also prognostic biomarkers. For example, we are now working in rectal cancer imaging biomarkers, and we have identified already some imaging biomarkers of the heterogeneity properties of the tumor that are telling us what patients will relapse. And therefore, we can undertake more aggressive treatments from the beginning and not simply the standard therapy. So it's helping also us to identify, for example, different branches in a clinical trial, right? What patients go to a treatment branch and what patients go to another one. So the biomarkers can help us in identifying also these questions. And finally, also to evaluate treatment response. They can be used to monitor if a specific drug is producing any change at a tissue level or at an organ level. So this is the main information that we can obtain from imaging biomarkers. But nowadays, there is a global concern, I would say, in the field of imaging biomarkers. And this concern is related to the reproducibility and the accuracy. if we go to most of the radiology meetings, we always have kind of final slide in most of the talks saying, okay, we need standardization, we need harmonization of imaging biomarkers. And it is true. We need nowadays to achieve a consensus between all the methodologies that we use to extract information from images. And for that, my co-founder and myself, my co-founder is Professor Marty Bonmati. He's a key opinion leader in the field of radiology. So we decided to promote a kind of model to extract imaging biomarkers. And for that, we defined a model which is established as the stepwise methodology. And we published also a book on this model, on how to measure images. And in this model, we try to minimize the degree of uncertainty in the measurement. And that means that we have to control the biases due to the use of different vendors in the image acquisition. That means that if we generate images using a machine from a vendor A or from vendor B or vendor C, we will get images with slightly different quality and different properties. So we have to cross-calibrate across vendors. And the processing and the algorithms can also introduce variability. And we also know how to apply algorithms that try to minimize the variability between the different methodologies that we can use. So that means that we have to work in consensus. For example, to apply always the same image filters or to apply always the same movement correction algorithms, and try to give importance to these algorithms and try to achieve a consensus in all of them, and also in the algorithms that we calculate. So as of now, although there is no consensus on metrology aspects of imaging biomarkers, we decided to, let's say, bring the requirements or the limitations from the blood test. So since there is no, let's say, standardization, we thought, okay, how can we, let's say, ensure that our measurement techniques that extract imaging biomarkers from CT, MRI, or PET imaging or X-ray, how can we validate them? How can we establish a methodology for validation? And we simply decided to work with the same requirements that are established right now for blood laboratories, for biological sample laboratories, and we decided to work with the European Medicines Agency regulation that establishes that for any bioanalytical test, the variability should be below 15%, and the accuracy compared to the ground truth should be also below 15% of relative error. So what is the highest level of accuracy that we, or the lowest level of accuracy that we accept for our imaging biomarkers? It is mainly the 15%. So for a biomarker to be validated, for an imaging biomarker to be validated, we require a variability below 15% on repeated analysis and an accuracy with an error below 15% compared to the ground truth, okay? So what are the therapeutic indications that are ideal for imaging versus blood? And I would say that the main indications for imaging biomarkers are those where we need to have a high resolution on a tissue and organ level. I mean, blood imaging biomarkers are simply giving us, blood biomarkers are simply giving us a number, I would say. And imaging biomarkers are resolved in space. That means that we can see where is the biomarker augmented. For example, in a liver, we can see where is a high fat concentration or where is a high iron concentration. In lungs, we can see where is the emphysema, where is the most aggressive cells in a tumor. So we have this space delineation that is so powerful, right? For example, for guiding biopsies, for example, for seeing where the tumor is developing the necrosis area, and I think this is the most important part, right? And let's speak now on, if there is no question, I think that there are no questions for the moment, right? Okay. So
Doesn't look like there's any questions at the
okay.
moment.
So let's move on to the concept of real-time clinical trial. This is a quite new concept. Okay. Why? Because as of now, we are quite used to have standard clinical trials where we collect all the data, and in the end of the clinical trial, the CRO performs all the analysis and provides the conclusions, and then a paper is published and so on. But we have developed a platform for centralizing the medical images in a clinical trial, and this platform has embedded all the quantitative image analysis algorithms that we have in our company. And this platform, for example, allows things like, as soon as we receive an MRI or CT study from the clinical trial that we are ongoing, this study is automatically analyzed using our algorithms, and several imaging biomarkers are extracted from the data. So we are converting the images to data, actually. And this data can be stored in the database and can be mined and can be used to extract early conclusions. For example, as soon as, imagine that we have a recruitment cohort objective for a clinical trial of 1,000 patients. So as soon as we get 100 patients, we can already see what is the relationship of the, for example, of the drug effect with the age, or what is the relationship of the drug effect with a specific toxicity imaging biomarker, for example, or what is the relationship with the age, or what is the distribution. So we give access to the sponsor and the principal investigator and the CRO and the super users of the clinical trial to this kind of powerful information that helps them to make early decisions. So for example, imagine that we have a recruitment population of 1,000 patients for a clinical trial, and as soon as we are in a 400 number of patients of 400, we can see if there's no difference in the treatment effect, for example, between different branches. Okay, so I think this is the real-time clinical trials concept. At any time of the study, we can enter into the platform, we can see the results of the analysis in all patients, and we can help to undertake decisions, right, related to the clinical trial. For example, decisions on continuation, let's incorporate more patients, decisions on stopping the clinical trial, or decisions on early publications on extremely relevant results, right? So this is the real-time clinical trial concept. So at any time, let's see how the drug is behaving, right, with our cohort. So what is the technology supporting it? Actually, the technology that we have developed is a cloud-based platform that allows the centralization and management of medical imaging examinations of any modality. So we are compatible with any nuclear medicine or radiological imaging modality. And it has a database which is based in Mongo. It's a very fast database that stores all the data of the clinical trial and the studies. Our platform allows to give access to the users from the different imaging examination centers, the different hospitals. And then we have other user roles for the managers of the clinical trial and the sponsor, right? The system is working right now in Microsoft Azure, which is our cloud provider. And the algorithms are mainly developed in Python and C++ and MATLAB languages. So we have like a server for the main database for the clinical trial. We have also servers for the computational analysis. And as soon as we have a new contract for a new clinical trial or a new study, create a new instance, we create a new private version of our platform for the clinical trial. So imagine that we are offering the service to a sponsor A or a CRO A. So we create a new space, for example, called sponsor.quiebeam.com or cro.quiebeam.com. And this space is private for the clinical trial, and then the hospitals and the centers for this specific trial can upload the studies there. And we give all the user accounts and access. In our company, we have the centralized quality check of all the examinations that are uploaded to the clinical trial. Also the quantification. We have radiologists also performing the standardized and traditional radiological reading. And in the end, we provide the full package of solutions for clinical trials, right? So this is more or less how are we working as a centralized advanced core lab. So if in case there is any doubt or concern on the technology, please ask us, right? But the algorithms that are running behind allow, for example, to as soon as we upload a brain study, we will get all the volume parameters, and if there is any lesion, and all the lesions quantified and the volumes quantified. For example, as soon as we upload a chest scan, we will get data on lungs, on bone quality, and a lot of biomarkers. As soon as, and etc., right? As soon as we upload, for example, a prostate MR examination, we get information on the prostate, and so on. So regarding the adaptive protocols and to make changes to the protocol in real time, I mean, I think that one of the most relevant values of this real-time clinical trials concept, thanks to imaging, as I was mentioning, we can have, or we can make early decisions on our clinical trial and make slight changes in those protocols. For example, if we see that there is no difference in let's say, a clinical endpoint like the relapse between two different treatment branches, but we have a difference in, for example, let's say, survival with a specific treatment, we can increase or we can decrease the number of patients in a specific groups of randomization, right? So we can make real-time modifications on the clinical trial, and I think this is important. Right. Okay. So let's speak about the quantitative image analysis specifically, right? So the quantitative image analysis is based mainly in is the process of extracting imaging biomarkers. And we understand that we have to perform quantification in all the images. Why? Because we know that even if we are in a clinical trial of cancer, right, and we are obtaining images from chest, abdomen, and pelvis, and we are looking for lesions or metastases, we have a lot of information that is hidden to the naked eye and that should be quantified. For example, in a chest, abdomen, and pelvis CT scan, even if we are looking for tumors or lesions, we can extract information on lung properties, we can extract information on vertebral body properties, we can extract information on visceral fat properties, and probably this information may be related to cancer later. So it's important that, although it's not in the, I would say, in the primary scope of the trial, that we perform this massive pooling of data from the medical imaging examination. And I think that's the value proposition. Everything that is measurable in the images should be measured. Everything that can be quantified from the images should be quantified. There is a famous statement from Lord Kelvin saying that if there is something that you are speaking about and you can measure it, then once you measure it, you start to know something about it. So unless we measure the images, we will not have the full spectrum or the full view or the full scope of what's happening in the disease. So our main motto in the company is that to measure is to know, and we have to know everything that's happening in the patient. And in order to know everything happening in the patient, we have to extract as much imaging biomarkers as possible, right? So let's think in the therapeutic indications of quantitative image analysis. I think that quantitative imaging biomarkers are so powerful because they are highly sensitive to small changes of treatment, right? We can monitor, for example, at some days after a specific treatment in a tumor to evaluate if the angiogenesis is changing. For example, we can see it at 7 days after treatment of a specific lesion or tumor. So early treatment response changes are one of the indications for treatment response monitoring using imaging biomarkers, right? And also the evaluation, for example, of radiotherapy efficacy combined with chemotherapy is useful, right? Also treatments related to stop brain degeneration or brain atrophy, treatments, for example, also for metabolic disorders like steatohepatitis. So the therapeutic indications that we are working with are mainly in 5 pillars. One of the pillars is neuro. We are working in atrophy, imaging biomarkers, and gray and white matter lesions, imaging biomarkers, and we work mainly with Alzheimer, Alzheimer disease, ALS, amyotrophic lateral sclerosis, also multiple sclerosis, and mainly also neurodegenerative diseases like Parkinson, where we have an increase in iron deposition. So, for example, in the field of neuro, we measure the volumes, we measure the atrophy, we measure the white matter lesions, and we measure, for example, the iron deposition in the brain because it's related to some diseases like Parkinson and amyotrophic lateral sclerosis. The second pillar is the pillar of oncology. In oncology, our value proposition is to perform kind of virtual biopsy of the tumor. So we quantify the angiogenesis of the tumor, we quantify the cellularity of the tumor, and we quantify the heterogeneity of the tumoral tissues. Okay, and we apply that to all solid tumors that we have nowadays in literature and in most clinical studies. And beyond solid tumors, we have a very specific solution for hematology diseases. We are working with multiple myeloma and lymphoma clinical studies, and we have been recently involved in a clinical trial in multiple myeloma, monitoring minimal residual disease. And for that, we have analyzed the PET-CT examinations post-treatment in order to evaluate residual disease that was not evident to the naked eye. And we have identified patients that have minimal residual disease using a quantitative image analysis of the PET-CT scan post-treatment. Regarding other indications, for example, we are working with steatohepatitis in liver, and this is very important because recently, in a quite important paper, we have demonstrated that steatohepatitis is related to the coexistence of not only fat, but iron in the liver tissue. So we have developed a method to simultaneously quantify iron and fat concentrations in liver, and it's having a great success because it's acting as a prognostic indicator of fibrosis and later cirrhosis in liver. And we are working therefore in the quantification of these fat and iron deposits. Another indication in another different area is COPD. We have a patented algorithm for the one-stop shop quantification of all chest properties in COPD. So we are quantifying lung volumes, lung emphysema, air trapping, coronary calcium, aorta and pulmonary vessels ratio. So many different properties that can be extracted from a chest CT scan. And we are currently involved in one of the biggest observational studies worldwide in COPD. And finally, we have the musculoskeletal indication. So we are very good in quantification of trabecular bone properties. We started developing the trabecular bone algorithms mainly for osteoporosis drugs. But suddenly we noticed that the algorithm had a very nice application in some diseases with osteolytic lesions, like, for example, also multiple myeloma. So we are also providing imaging biomarkers of trabecular bone destruction and trabecular bone microarchitecture. And why we do this? Mainly because densitometry, that was the gold standard nowadays for the osteoporosis diagnosis, we have seen that there are around 40% of patients of osteoporosis that have, I would say, normal densitometry values that are breaking the bones, actually, are breaking the femoral neck. And there is a nice paper that was published some years ago saying, what was your fracture score the day before your bone fracture? So we have noticed that, the standard techniques like densitometry are not catching a lot of non-traumatic bone fractures that are happening. And we think that the problem is in the microarchitecture. We are not quantifying microarchitecture in daily routine. And we have developed an algorithm to quantify the bone microarchitecture from MRI, from CTs, and from cone beam CT scans nowadays. So this is another indication, right? And the technology that we require for that nowadays is mainly related to high performance computing hardware, but is providing this service in the cloud. So there is no need to have, servers or hardware or high performance computing resources at home or at, let's say, at our offices. What we need is just to ask for the service. I think that we are in the era of services, and Keybeam is providing these services for all the clinical trials and sponsors, right? So let's go to the one of the most important parts, according to FDA, for medical imaging core labs, right? And one of the most important parts is to have an imaging review charter. Keybeam, as advanced quantitative imaging core lab for clinical trials, we have to have, let's say, a quality management system. And one of the big pieces of this quality management system is the imaging review charter, right? Obviously, the imaging review charter in the end is suited or it's customized for every clinical trial or clinical study. But what FDA is telling us is that we have to incorporate within our companies an imaging review charter, let's say a document or protocol describing how the images are received in our laboratory, in our virtual core laboratory, how the images are managed, what are the quality parameters, how we perform the quality check reports, how the images are analyzed, using which techniques, how the images are read by radiologists or nuclear medicine specialists. Are they, for example, double read? Are we using two readers per imaging? Sorry. How we relate the images to the clinical endpoints. So nowadays, the imaging review charter is, as I was mentioning, a very important part of a clinical trial that is involving medical imaging. And the sponsors, the CROs, should be asking the core labs, the imaging core labs, for this kind of imaging review charter, and obviously the quality management system. So it would be interesting to know for all the attendees what type of company is best describing you. And it would be nice if you can vote on the poll, and it would be interesting for me in order to, to address the conversation. Okay. Okay, we have site, we have CRO, we have other. Okay. Okay, I don't know if everyone submitted the vote to the poll.
Yeah, we have another that said industry vendor.
industry vendor.
Okay. Okay, great. So I think this is the importance of the imaging review charter, right? So in the end, what we are providing to clinical trials is a full package of services, right? Because, and this is important, we, as company, we started to offer the advanced imaging solutions. And in the end, we noticed that we cannot offer the advanced imaging solutions alone. We have to provide the full solution involving all the services that are related with images in a clinical trial. So for example, we are providing right now the design of the image acquisition protocols. So we even go to the centers and we train the centers on how to acquire the images, and we have a specialized team within the company on doing that. Once the images are generated, we train the centers on how they have to upload the images. So that means that we do a training. Once, for example, we have received the images, we do the quality check analysis. If there is any trouble, we generate also the queries to the centers, and we try to mediate between the center and the clinical trial, just helping the centers to solve the imaging queries and trying to improve also the quality of the images. We perform the radiological reading and the nuclear medicine reading, and we perform also the quantitative image analysis. And finally, we are providing periodic reportings on the results of the clinical trial to the sponsor, the CRO, and the principal investigators of the trial. So the full package of service that we are offering there. So this was more or less all the topics I wanted to share with all of you. And I am more than welcome to your questions right now. I think we can foster an interesting discussion. Thank you.
All right. Thank you, Angel.
Thank you, Angel.
And I hear an echo, so let me mute you.
Yeah.
So what's interesting for me about this
is, as a CRO, the real-time and adaptive capabilities of this technology. I mean, this is obviously something that's going to play a crucial role in making trials more efficient in the future. And it's going to be something that's definitely going to be coming into play. It already is. I mean, Angel can tell you about that. And if you follow us on his podcast, Keybeam blog, as well as keybeamblog.com, you'll see some of the contents and video interviews that we put together to talk about how this stuff is already affecting the clinical research landscape, and it's only going to become more so as we get into the next decade. So this is definitely, you guys are all at the forefront of brand new technology that, quite honestly, the industry has desperately needed for quite some time, and now the technology is actually making it possible for us to do real world adaptive trials. So we can change a protocol on the fly when needed based on these results from these imaging biomarkers. So thank you everyone for attending. Again, this is Angel's email address. If you go to keybeam.com, that's the website. If you go to keybeamblog.com, that is the blog. And make sure you subscribe to Keybeam's YouTube channel and go like the Facebook page as well. Everyone will get this replay link sent to them, and it will also go on the Facebook and YouTube and podcast for Keybeam. So if there are not any other questions, I think we can end this webinar. And again, email all questions you have to angel@keybeam.com, or you can send them to me if you prefer. And most already have my email, but it's dan@theclinicaltrialsguru.com. So we hope to do more of these with Angel. I find it fascinating, actually, and for me it's timely as we're building a CRO. This is all pertinent information for me, and I hope it is for you as well. So thank you, everyone, again, and thank you, Angel. I'm not able to unmute you unless you're able to talk.
Thank you.
Okay, there you go.
Yeah.
Yep, and we look forward to doing more of these with the audience. So we'll eventually cover other topics that the audience finds interesting or would like to know more about, and just correspond with us whenever you would like. Thank you again, Angel.
Thanks to you, and thanks to you all. Very happy to share this information with all of you, and I think this is not the future, but the present of clinical trials, right? The future is now.
The future is now, correct. And with that being said, good afternoon to all of you, and good evening to you, Angel, over there in Spain. Have a good rest of the day, and we'll catch you all later. Thank you very much.
Thank you. Bye. Thank you. Stay in contact.